Right, welcome back. This is the continuation of Chapter 5. We're looking at Part 3, and what we're going to discuss is really a kind of a follow on from the last time, but I just wanted to convey it's amazing that the efficiency, I guess, having millions of years to perfect it, but the photosynthesis is, I would say, nearly 100% efficient. And I'm going to show you how this conclusion is derived, but there there are quantum mechanical effects, but the key point of controlling the efficiency boils down to something we already talked about in class, is about percent hydrogen, pH. And as you'll see, as the process of photosynthesis occurs, and you're building that battery effect across the membrane, there's a control mechanism to make sure that it doesn't overcharge. In other words, the battery doesn't overcharge. In other words, the pH doesn't get too low. And so let's talk about how this comes about. So I went ahead and I wanted to share with you how the research was done. If you're slightly interested in maybe graduate school at some point, this is some research that was done at a university, and you get to see the graduate students, and it's just a piece of research that I wanted to share with you. And again, it's quite amazing. So his group, you'll see in a moment the professor, but Graham Fleming, he's a physical chemist, pea chemist, which is I say pea chemistry and threw up just a little bit. I remember that being in taking that course in graduate school. It was a course to remember. It was a weeder course. If you don't know what a weeder course is, you're weeding out your grass. They're trying to get rid of those who don't really belong in graduate school. So they just make this course really, really tough. And so anyhow, it's a fond memory, I guess you can say. Not, but anyhow, he got his Graham Fleming got some grant monies from the U.S. Department of Energy, and the University of California at Berkeley is where the study was done. And a group, as you can see the names here, but let me show you. So here's the professor and the graduate students. They're very common. I would highly recommend, if you have any interest at all, go over to NC State or UNC, or whatever your favorite, or Duke for that matter. Walk around campus, whatever, when we get back to normal life. That Walk around to some of the graduate schools and some of the graduate programs and talk to the graduate students. See what they're doing. If you don't leave excited, or at least really curious about wanting to pursue in some higher education, I tell you, just to visit, and I tell you, graduate students love to talk about their research. Now, a lot of times you just go to the department head of a particular school and say that you'd like to visit some of the graduate programs, just to get an idea. They will let you do it. It's really important to open your vistas and kick the tires, let's say. Get out there, see what's out there, see what you might be interested in. Half the time I hear from my students, so what's your goal? Well, I don't know. Well, obviously it's very hard to know because you haven't explored. A lot of students just haven't had the opportunity to go out and explore some of the avenues of things to do. It might interest you. You never know. There may be things just right under your nose. You say, wow, that's really cool, and get energized, and before you know it, you may be pursuing that. So anyhow, here's, of course, they're looking at quantum entanglement, and those things are really, really cool concepts. But they bring this down to just just straightforward. Look at all the mirrors, reflectometers, and all these things to be able to redirect light so they can study, and I'm sure they have some good cameras and all sorts of things, and it's a phenomenal study. So what do they do? Let me go over one. The pigment binding protein CP29, which I pulled out of the PDB data bank and I printed it. It's a very interesting protein. It's one of the minor light harvesting proteins in green plants, and it's identified as a valve that permits or blocks uh, the critical release for excess solar energy. In other words, it's a little flap that moves, and when the pH gets down to a point, it changes its shape and closes that opening, so the sunlight doesn't get in. And it's phenomenal. And so when it utilizes some of the, the pH starts to increase because it's not being replenished or the battery's not charging, eventually the pH will change, and then the CP29 protein will change its shape. And as a result, it is a built-in automatic. Once the protein's been manufactured, it goes in place, phenomenal, and then behaves based on that pH within the charging of the battery across the membrane with the cracking of the water. So this tells you, though, in order not to overcharge the battery, in other words, we don't want to damage the cell, it's regulated so tightly that the efficiency it's got to be approaching 100%. It's just amazing. So as the pH change, what's nice about this, it is a reversible type process. So when the pH changes, the CP29 changes its shape, and it's just back and forth. Here's a look of that little CP29 protein, and you can see 8 chlorophylls. Now remember that what the chlorophylls do, remember, they're integral to that excuse me, the sending of the pitch. In other words, it's when the light hits it, ping, remember, just the right frequency, and it resonates. And if you block that so that the chlorophyll can't get pinged, it's not going to resonate. And what's it going to do when the chlorophyll resonates? It's going through and it's cracking water. And that's where we get the hydrogen ions, and hydrogen being part of the percent hydrogen. So the more you produce, the lower the pH, and hence the effect of this protein. And I just love these things. It is just amazing. Now this is a spiral form. I could do a solids model of this, which I've done, but it just shows the influence of the atoms that of the amino acids that make up this particular protein, and how it holds in place all of these chlorophyll molecules. And it's stunning to me that nature is to be able to see the details going on. It's very rewarding. So anyhow, I thought you'd like to see that. Okay, now photosynthesis, capturing the energy of sunlight, is used to make sugar. Now we're going to talk about the synthesis part. I've gone over the photosynthesis part where the sunlight and we crack water to get the energy, and you've already been through that. And now we're going to talk about a part that ultimately what we're doing is making the sugar. So everyone that's involved in the team. So think of it as the day crew and the night crew. The day crew of all the enzymes and everything, or the if you use the business analogy, you hire the staff that do all the things that ultimately at the end of the day creates the ATP. And now they clock out, and then the night crew clocks in. Of course, they're all wearing Calvin Klein clothes. It's a different shift. It's just a different thing. So now we're making sugar, which is refined. We make cookies with the sugars and all that. It's just a different deal. So I use that analogy, and it's all right to do those sorts of things because it helps trigger memory, and so the Kelvin cycle. But the idea is, this is the night crew, and what they're doing now is taking the ATP, the NADPH, and carbon dioxide from the atmosphere. In other words, that's where we're getting carbon. So we're making sugar molecules, and part of the carbon is coming right out of the air. We fix it. They call it fixation. Right out of the air. And so the carbon dioxide, what we breathe out, and then I guess you can imagine at the end of the day, having sugar molecules that we can store or the plant stores, and we're off to the races. So there is a protein, and I wish I had you in class. I printed the protein Rubisco. Rubisco is the most abundant protein on the planet, and I'll talk more about that. But Rubisco plucks carbon atoms, so it is the molecule, the protein molecule. Now, it's a multi-enzyme. In other words, this is just one of many things, but it's the most abundant protein on the planet. And you can imagine how important it is to fix the CO2, get that out of the atmosphere, and then have our plant friends pump oxygen. We need the oxygen. We breathe out the CO2. What a match made in heaven. Can you imagine that? So here's Rubisco, carbon dioxide, and part of the enzymatic activity, which what enzymes do is they reduce the amount of energy required to do some task. And in this case, we're building an organic molecule, glucose, and Rubisco is taking the carbon dioxide, docking it, and putting it in such close proximity to the growing organic molecule that using ATP, that chemical bond energy, we can now, and NADPH, we can now make sugar. And there's cycles that goes on. The gentleman that worked this Kelvin cycle out, what a phenomenal detail. I bet he was really, really intrigued with what he discovered, because this is just amazing. But sugar creation, so the night crew is totally different. Don't need photosynthesis. All the output of photosynthesis during the day crew is ATP and NADPH. So chemical bond energy, the phosphate bond energy, now drives the ability to move energy from the organic molecule and the carbon dioxide, and we implant that chemical bond energy in the form of sugar. And I hope everyone followed me on that. So the processes in the Kelvin cycle occur in 3 steps. Enzyme and coenzymes are recycled. Rubisco, the most abundant protein on Earth, ADP and NADP. So we're going to go through an oxidation reduction type of reaction. We've talked about the redox type of equation, where we're filling or emptying a seat. So the Kelvin cycle takes CO2 and builds it into molecules of sugar. Now, what is Rubisco? Well, the reason I like to say it Rubisco, look at the mouthful of the name of this enzyme. And I love how the chemists name these things, because it tells you what it does. But it takes ribulose 1,5, bis, in other words, got 2 phosphates, carboxylase. It's any word with ASE at the end is an enzyme. So what does it do? It takes carboxyl groups, or an oxygenase. It deals with oxygen as an enzyme activity. So it moves oxygen and CO2. So the carboxyl, the CO portion, and oxygenase. So Rubisco is the primary catalyst for photosynthesis and the basic means by which living creatures. Now, this is out of your textbook. And what I'd like to say, it's a principal catalyst for the dark reaction within photosynthesis, the night crew, the Kelvin Klein dressed staff that's working to make sugar, or cookies, as you may want to say it. So here's Rubisco, and it has several large chain dimers. And you can see, I don't know if you can see it clearly, but there's a gray area and a white area, and then the small chains, the blue and orange. It's quite an amazing protein. As I say, I printed this. It's just an intriguing protein to look at. It's crucial for carbon fixation. It's a source of energy, so it does the task of transforming energy in the form of chemical bond energy from the ATP, that the ATP syntases are spinning, all the battery charging, all that discussion, ultimately making that ATP is now utilized or picked apart by Rubisco to take the CO2 out of the air and then transform the chemical bond energy of ATP into ADP using that phosphate chemical bond energy. And we can now build these glucose molecules. So if I take that kind of generic chain and now give you a little bit of meat on the structures, here's the ribulose 5-phosphate. Hey, look at that! Isn't that amazing? The enzyme mentioned something about ribulose 1,5-phosphate or bis-phosphate. So it takes ribulose 5-phosphate with ATP and converts it to ribulose 1,5-bisphosphate. Now you can see why the name of the enzyme of Rubisco is what it is. And adding carbon dioxide now, because we've got the molecule in this very strange state that is willing to adhere to this carbon dioxide, because the molecule can accept it in that it's opened up just a little bit under stress from the ATP, the chemical bond energy, allowing now the carbon dioxide to come in and bind, and to the 3-phosphoglycerate formed as a result. And it's funny how nature does these sorts of things. We build these longer chains and we kind of split them, and so that they run out to various different pathways. But a 3-phosphoglycerate, an ATP, again, to get the 1,3-bisphosphoglycerate, using NADPH, that hydrogen, that's that chemical bond energy with that little extra hydrogen, we get glyceraldehyde 3-phosphate. So what we have are or half of the glucose molecule being generated, and so 2 rounds of the of this Kelvin cycle, we can build a glucose molecule, and it's just phenomenal. So the synthesis part, the dark or nighttime crew part of photosynthesis, is the Kelvin cycle. During this phase, carbon from CO2 in the atmosphere is attached to molecules in the stroma. So if you remember, all the ATP was being dumped out from the inside of the bag within the bag. The inner bag is that battery effect, and it spins the turbine of the ATP synthase, and then when it makes the ATP, it dumps it right into the milieu, or the outside of that inner part. And they refer to that again as the stroma. So sugars are built and molecules are regenerated to be used again and again. The fixing, the building, and the regeneration process consume energy from ATP and NADPH, which were all made during the day cycle. In other words, the day crew did all that and allows now the night crew to integrate it to make sugar. Rubisco, being the most predominant protein in the world, it's also one of the most flexible proteins. It can operate under, we talk about enzymes as temperature, sensitivity, and pH sensitivity, and all these things. It does. We do tend to run into a little bit of limitation because we're really pushing the operational, the width of all the operation conditions, the temperatures and everything in the environment. And the weakness of this enzyme is the cause of photorespiration, which is such that healthy leaves in bright light may have 0 net carbon fixation when the ratio reaches a threshold in which oxygen is fixed instead of carbon. And this is a temperature. In other words, it shifts the shape of the Rubisco so that it can't convert as well, and it loses efficiency. So the plant has to think about that, and through evolution, believe it or not, the plant made some adaptations utilizing the Rubisco. So let's talk about that. So in Sudan, Ethiopia, these are the world's regions with the course starvation. It's just terrible. It's the hottest and driest climates. It's almost inhospitable to humans. But the climate changes really present difficult challenges for agriculture. And it's really interesting, though, how plants actually have evolved to handle this. So stomata, which means opening, in Latin it means mouth, and the end is the person, place, or thing. So it is a thing, and it's a sort of an opening for gas exchange. So we have this ability now to open and close this sort of mouth in the plant. These openings are primarily sites for the gas exchange of plants. Carbon dioxide for photosynthesis opens or goes through these openings. Well, when open, the stomata allows water to evaporate, which is if you're in a really desolate environment, it's hot and all that, closing the stomata would be a good solution because you're not allowing evaporation. However, you get too much water evaporation, or you try to stop the water to evaporate, the stomata shut, oxygen from the photoreaction cannot be released from the chloroplast and carbon dioxide can't enter. So, you can't just shut the door. So if there's no carbon molecules for the sugar production, the Kelvin cycle tries to fix the carbon, but instead only finds oxygen. And so the plant goes to a standstill. So what does it do? Well, as I mentioned already, there's a slight adaptation that goes on, and we can see we have names. So the normal process is the C3 photosynthesis. There's one now called the C4, and gases are exchanged through a slightly open stomata, so it's not wide open, slightly open. And so it cuts back or throttles back the amount that gets in. So it's sort of that happy medium between, well, we're not going to allow full water to get out or evaporation, so we kind of live in the center where we can kind of just make it by. And so the genetic or the adaptation is this sort of partial opening, and the carbon uptake requires additional energy because it's got to run that engine a few extra times. It occurs in about 1%. It minimizes water loss in these warm climates, but it does require more energy. But a little bit, the old adage, you got to pay to play. If you're going to be in these environments and survive, which some plants can't do it, well, it's better to do something at a cost than not have anything at all. So there's the C4. Now, CAM photosynthesis. Gases are exchanged at night through open stomata. Carbon uptake requires additional energy, and then the stomata closes during the day. So at night it's only open and it's closed during the day so we can make the sugar. And this occurs in 3 to 4% of the plants. Water loss is minimized in warm climates, but the problem is that we have to kind of go through that day-night cycle in order to exchange the gases. So it's slow growing. But again, either you're dead or you grow slower, and it costs a little bit more. That's a pretty good fair trade, and you can see cacti down here is the type. So the added CAM, you think of it like the camera, the day and night camera. I don't know how you're going to remember, but think of it as the CAM process, where at night it does the exchanges and it kind of builds up, so you can imagine the intake of this area is a little bit bigger, and during the day the stomata closed, but it's got all the things that it needs to go through. So isn't that interesting? The day cycle where it closes and does the photosynthesis, but it makes sugar during that time and is trying to minimize water loss. So you can see the C3 and C4, and we get C4, no photorespiration, full sun, moderate use, and that sort of thing. You can see the changes in its efficiency and performance. But again, depending on the environment that you're at, you can see the distribution around the equator. Of course, we tend to have the C4. Otherwise, you won't have them. And there are some that can try to survive in that environment, but at the conditions where it's really hot, you're not going to see it. So C4 and CAM photosynthesis evolved because it made it possible for plants to grow better in the world's hot and dry regions. The enzyme, the Rubisco, still works, but it had to to modify because of the temperature. That's why things also grow slowly. I mentioned it as the amount is being gauged, limited to the exchanges of the CO2 and oxygen, which is true, but also the Rubisco with the higher temperatures is not as efficient as an enzyme. So, but it still works and it gets the job done, which is amazing. So, C4 and CAM photosynthesis are evolutionary adaptations, a slightly modification of the standard C3, and so I wanted to point that out. So the food is fuel, so sugar. So this part of the presentation, we're going to talk about what, of course, uses sugar. And, of course, all of the energy sources that we get, the fuel comes from the sun, all of it. So cellular respiration, the big picture. And if we look at cellular respiration, in other words, we take the glucose. So this is now changing the track of our philosophy and thinking. This is a process now where we take the energy from what was made during the night process of photosynthesis, i.e. the cookies or the glucose. Now, through the breaking down, glycolysis, lysis means the breakdown of glucose into pyruvate molecules, and we transform energy and make ATP. Isn't that amazing? We're able to recover the chemical bond energy that we put in the glucose and retrieve that using the broken down components, releasing carbon dioxide, the pyruvate going to acetyl-CoA, and again the Krebs cycle now giving off carbon dioxide, NADH and FADH2. Isn't that interesting that ATP is being formed because ultimately we're splitting water again through the high energy brought to us all the way from the glucose being shifted down through the electron transport chain, because once we modify in the chemistry of the Krebs cycle, we can take those electrons and charge the battery to get ATP synthetases to spin in a totally different mechanism, but it doesn't matter from your perspective. It's still charging the battery, but it was the residual energy from cracking water way back originally, and now we use the chemical bond energy that we used obtained in the ATP to now do it again and form ATP from the chemical bond energy in glucose. And it's sort of amazing that it's almost identical. So glucose through glycolysis, and then we have a mitochondria, and we're giving off CO2, so it's respiration, so we're giving that off, but we're forming ATP through the electron transport chain, and the net result is water and forming ATP, so we can now charge the energy of our systems. So respiration is the energy is released from the chemical bonds of food molecules, i.e. sugar, stored in the bonds of ATP, that chemical bond energy of that phosphate group, into usable forms that goes and does the work that we've been talking about. So here's the big picture again. Through the cellular respiration, we're breathing the oxygen and using the sugar, and we're harvesting now the chemical bond energy. We give off the CO2, so we put that back out in the atmosphere. We make ATP, and we're building water as a result of that. And so it's sort of the opposite from the photosynthesis that we've talked about, which should not be a shock because when we're breaking that down. But the net result of making the ATP is that we're charging the battery again with the hydrogens created through the process of taking the sugar and converting it and handling those high energy hydrogens in the formation of water, and we get the ATP. And it's just amazing. It's like everything that we talked about in reverse. So living organisms extract energy through a process called cellular respiration, and the high energy bonds of sugars and other energy rich molecules are broken, releasing energy that went into creating them. So it's just consistent with everything we talked about. The cell captures the food molecules stored in the energy bonds of ATP, transforms that, and fuel molecules oxygen, and it yields ATP by spinning ATP synthase by charging with the hydrogens. And to handle those hydrogen molecules, we bind it with oxygen, and carbon dioxide is released. So the water is produced ultimately at the end of the day from that process. So the first step is cellular respiration. I just wanted to kind of step through just to review using glycolysis and adding a little bit more detail. So in order to get the ball rolling, we had to expend some ATP energy to get the molecule unstable and broken and producing water, and we get now acquiring some energy in the process of this chemistry. So it's very efficient, but that's not the main win in terms of forming ATP. We're going through this process to create and charge the batteries, and so once we do that in the electron transport chain, look at the win, how many ATP we get, 34, and it's because we're spinning the ATP synthases by releasing all these hydrogens to change that pH. Now your book doesn't go into the detail about that, but I assure you that's exactly what's going on, and we get the big win of those spinning ATP synthases to get the big win on the ATP. So glycolysis is the initial phase in which all living organisms start to harvest that energy, and it occurs in the cell cytoplasm using energy from breaking chemical bonds. And so that's sort of in review that we talked about. So at the end of the day, for the glycolysis, we get the pyruvates and then coenzyme A, and this is now the ready form to start getting the big win of making the hydrogens that we need to charge the ATP synthases by charging the batteries. So as we break down the pyruvate or the glucose into the pyruvates and having some chemical manipulations that occur from that, the acetyl-CoA group kind of plugs into this merry-go-round, and we're building the NADHs. The carbon dioxide is released. We have a 4-carbon molecule. We have a little bit of ATP made, but look what we're doing is we're getting the FADHs and the NADHs, and that goes to the electron transport chain. So it's those hydrogens that are going to be released that's going to change that pH, or have those hydrogens charging that battery to spin that ATP synthase. So all of this write-up I don't want you to memorize, but if you wanted to know some of the detail, you can go through that. But I'm good to say that ultimately what we're doing is building up the hydrogens here and here in order now to charge that battery. So ultimately the payoff from the Krebs cycle, which is the big win, is the ability now to make the pH change for the charging of the batteries. And then we build, so following the 6 carbon atoms from the photosynthesis through cellular respiration, is that we take the 6 and we break it down by removing this one of the CO2s and creating the pyruvates, and we get 2 molecules of CO2 per turn of the citric acid cycle. So the huge amount of additional energy can be harvested by cells after glycolysis. So we're taking now the glucose, breaking it down, harvesting the chemical bond energy, and in a form that in the process of breaking that chemical bond energy, we're getting the hydrogens on those carrier molecules, the FAD and NADPHs, so that that hydrogen can now be used to charge that ATP synthase to spin and make ATP in a big win. So next time we're going to go through more of the electron transport chain, and just to kind of reiterate everything I talked about in case you didn't quite follow me. Usually my students do. The aha moment usually occurs in part 3, but I'll drive it home on part 4 next time. So stay well.